a novel automated high-throughput plasma protein biomarker enrichment protocol. Plasma protein biomarker enrichment is necessary to increase the dynamic range of plasma biomarker identification in an LC-MS bottom-up analysis. As you all know, most of the plasma is made up by redundant proteins that don't give you anything in information, such as albumin, immunoglobulins, etc. The problem with depletion, if you do this by affinity or other means, is that there's an unpredictable co-depletion of biomarkers due to complex formation, and thereby most people don't like this. Also, these workflows are not easily automatable, and they are serial. That means it's single columns or single tubes or a serial of columns that you have to go over. We looked for a solution by disrupting and dissociating protein complexes as they are coming onto the matrix, thereby disrupting co-depletion. And we do this by adaptive focused acoustics, or AFA, and I will explain that to you shortly. So physically, we subtract only targeted, unwanted, redundant species and leave the protein biomarkers alone, thereby decreasing co-depletion of them, increase the sensitivity of their detection, and increase reproducibility and repeatability of these processes. Everything is done in a 96 well plate, and keep that in mind. So whenever I talk about 60 minutes or whatever protocol, it means 60 minutes are 96 samples in this case, all at once. And also, it is directly compatible with downstream trypsin enrichment, so there is no exchange of buffers, etc. Adaptive focused acoustics, you will hear that many times during the talk. Some of you know it, but it is a precisely controlled energy transfer of sonic energy into a focal zone in the vessel. And you will see that basically you can control all of the aspects of this energy transfer. You can control the wattage, which is the amplitude A in the lower little graph there. You can control the time, how long a signal or how long a transfer will be, like T1, and you can control the pauses in between, which is basically a duty factor. And thereby you can control very precisely how much energy goes into the vessel, how it will be delivered, how the vessel will react to it, and the inside of the vessel will always stay isothermal. That's very important. You can control it by the water bath temperature. The consumable is on the right-hand side. It's a 96 well plate, TBX plate. You see the filling inside with the matrix of a typical depletion matrix and the plasma on top of that. And then on the right-hand side, you see the instrumentation, which is the LE220 series and the R230 series of Covaris. And the R230 can be set on a liquid handling deck, but the other one, the LE220 series, is also accessible by liquid handler arms or robotic arms. Here is the adaptive focused acoustic process, and it's the same. It's the matrix basically in a tube. We have little matrix in there to show the mixing. It's on the bottom, and I will start the video now. You will see in a microsecond how fast it is, and you saw that now, how fast it is getting mixed. We can do the mixing in a continuous mode. That means the energy is on at all of the time. In a pulsing mode, we can pulse it, basically on, off, on, off, on, off. And we can repeatedly scan this by mimicking the pulsing, repeatedly scanning it over a line transducer, thereby treating all 8 samples in a column repeatedly. And you can do this as long as you want. Repeating 100-fold, you can repeat 30 times, you can repeat only once, depending on what you want. The workflows that we employed here are the pure proteome EMD millipore plasma depletion kit and the high select top 14 thermal kit. Take a look at the one that is circled in yellow, the pure proteome for now. This one is on the left-hand side is the workflow. The protocol, as it is described, requires a pretty high volume, 1:64 dilution of the plasma. After that, you need to ultrafiltrate it in order to keep it into a meaningful concentration. You don't need to do that with AFA. That's the blue workflow. AFA is a 96 well. We have 50 microliters of total volume there, 2 microliters of plasma only. The depleted plasma is then getting treated over PCA to normalize the concentration, and then we handed it over to our collaborators at the UMass protein core, the proteomics core, where they did the TMT multiplexing, in this case for the pure proteome kit. and went through the LCMS analysis, which is explained at the bottom of the slide. If you have any questions, we can do that in the question and answer section. LFQ, a label-free quantification, was done with a high-select top 14. I will come to that after explaining to you the data from the pure proteome kit. Here is the depletion shown. So it is albumin and immune globulin that is depleted by this kit. The undepleted plasma is normalized to 1. Basically, the rotator is the kit protocol. It depletes it. And then you see also that the AFA treatment, when you scan the plate over the transducer in increasing amount of times, 35-fold, 70 times, 140 times, etc., you also deplete the plasma. The STS gel shows the depleted volumes, and down at the bottom, which is important, you see basically that the kit protocol depletes plasma 5.4-fold, whereas the Covaris scanning protocol depletes it about 4-fold up to 5-fold at the AFA 500, but that's a long time, and we would like to keep it between 140 and 280, and I'll show you in a minute why. Total energy impacts biomarker enrichment factor, and you will basically see that AFA is absolutely required to increase the biomarkers in the depleted plasma. On the left lower hand, the plot shows you raw plasma signal intensity versus pure proteome plasma signal intensity. This is the kit protocol, and you see, yes, there is an enrichment of plasma biomarkers, but if you apply 140 scans in the plate format with AFA 140, which is the middle one, middle plot, you will see the dramatic increase in biomarker enrichment or retention, and even more when you do double the scan time to 280 or scan numbers to 280. So really, you see an increased amount of biomarker retention. In the next slide, it's even more dramatically shown, where you can see basically that and I showed just the 280 scans here. The pure proteome protocol has a modest enrichment. I wouldn't even call it necessarily enrichment. For most of them, it's basically the same than in raw plasma. It's basically not totally co-depleted, whereas in AFA-enhanced protocol, you see basically in 1 hour the 96 well plate is basically so treated that you have a real enrichment of the biomarkers there. These are 130 biomarkers that are non-redundant, and they're plotted next to each other. There is none of them that is missing, so we have no biomarkers that fall below that were there in raw plasma or in the Cullen protocol, and they're not there in the AFA. That's very important. So when you look at certain proteins that require AFA treatment, these are proteins that are selected randomly, basically, that in enriched plasma, when you do the Cullen protocol or the magnetic bead protocol with a passive mixing, you actually see no enrichment. It's basically raw plasma is not number one, is 1, and then you see basically that they are not at all enriched beyond the 1, whereas when you do 140 scans or 280 scans, you see a dramatic enrichment of those biomarkers, and that's exactly what we want. Now switch gears to the schematic workflow of the high-select top 14 thermal kit. So we did basically nothing else than comparing the single column protocol as the manufacturer ones with the AFA protocol. In this case, we switched a little bit, we modified a little bit the AFA protocol. We had only 40 microliters of total volume in there and 1 microliter of plasma. And this was done with a label-free mass spec. Here you see the high-select top abundant proteins as they are basically enriched over this column. Raw plasma 236, total proteins identified that are non-redundant. AFA age-treated 305, and age-treated means it's a pretty high energy, nice mixing, nice dissociation of proteins. AFA-treated is a little bit of a lower energy. You see they're about the same, but then you see that the depletion, the total depletion of the proteins is much, much better when you do AFA-age. Only 280 micrograms left of the protein versus 1800 in the raw plasma and 300 of the Cullen kit, which is pretty good, but the AFA-treated low energy has a little bit higher. So that was good news. Then we wanted to look at how do these proteins that are targeted for depletion, in this case 14 different ones, how do they fare when you do AFA treatment? Are they better bound? Are they not at all changed with AFA, and only the biomarkers are enriched? But in this case, you really see that some of these proteins benefit from AFA as well. Albumin, modest increasement. That's the top one. But then when you look at IgG, for example, and IgM, there's an increase of tenfold in some cases for IgM, even more, and IgG when you treat with AFA. And that's very important. AFA high seems to really increase the binding of these to the matrix. And then when you go down to fibrinogen, haptoglobin, transferrin, similar increases that are dramatic. And we think that is because it dissociates protein complexes better binding. Alpha-lipoprotein didn't work at all, and this may be something that is intrinsic to the kit because it didn't work for the column kit as well, non-modified protocol. Antitrypsin had a modest, almost no increase in depletion. So that is something that is an outlier. We don't know how to explain that. But overall, better binding of the targeted proteins. Proteins that are identified only upon AFA treatment, that means you have them only enriched in the AFA-treated plasma, but not in the column-depleted plasma, are listed on this page. And they're pretty much the ones that are, you identify in addition to the raw plasma. They are not showing up in the column, so that means they need AFA in order to be depleted and in order to be showing up, and not to be depleted, sorry, and showing up in the enriched plasma. Summary: 2 kits were selected and modified to work on a 96-well format. They are by themselves not easy automatable, so you need to modify that. AFA dramatically increased the retention of biomarkers, even beyond the predicted enrichment factor. We normalize plasma by dissociating complexes, and that's exactly what it is. The AFA dissociates proteins from each other and prevents co-binding. We need to look for more previously characterized plasma samples. We have 1 collaborations. If you have plasma samples that you want to be looked at, please contact me. And we want to validate other depletion resins. Our collaborators, I thank Jason Evans, Director of the Proteomics Core at the University of Massachusetts, and Rachel Murray, who did all of the experimentation down there, and of course Patrick McCarthy, who did all of the work at Covaris here. Thank you.